Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about colony hybridization. Colony hybridization is a simple but powerful technique useful for screening gene library. In recombinant DNA technology, after construction of the recombinant DNA, the recombinant DNA is introduced into a suitable host, for example, E. coli and yeast. After introduction of recombinant DNA, it is necessary to select the bacterial cell that contain the recombinant DNA. By colony hybridization, one can easily select the bacterial cells that contain the recombinant DNA. Bacterial cells that contain the recombinant DNA can be selected and separated by the technique of colony hybridization. Colony hybridization is a blood analysis technique in which bacterial cells are transferred from a solid nutrient medium to a nitrocellulose membrane for further analysis. The technique of colony hybridization was developed by Michael Gromstein and David S. Hognes in 1975. So now let us discuss about the various steps that are involved in the process of colony hybridization. In colony hybridization, the first step is preparation of the master plate. For preparation of the master plate, a small amount of bacterial suspension is taken and inoculated on a solid nutrient medium. After inoculating a small amount of bacterial suspension on other nutrient medium, it will give rise to several colonies. So this is about the preparation of the master plate. Master plate is a agar nutrient plate that contains several bacterial colonies. That is the master plate. After preparation of master plate, next step is formation of replica. That means we have to create a replica of the bacterial colonies that are present in the master plate. For formation of replica, take a nitrocellulose membrane that is already sterilized. Take a sterilized nitrocellulose membrane and place the nitrocellulose membrane on the surface of the master plate and press it uniformly so that there will be impression of the master plate on the nitrocellulose membrane. So by uniformly pressing the nitrocellulose filter on the master plate, we can develop an imprint of the bacterial colonies that are present in the master plate. After preparation of replica, next step is treatment with detergent and treatment with sodium hydroxide. So this is the nitrocellulose membrane, and the nitrocellulose membrane having several bacterial colonies. We want to identify the bacterial colonies that will contain the specific gene or that will contain the recombinant DNA. So for that, this nitrocellulose membrane which contain the bacterial colonies is treated with sodium dodecyl sulfate. By treating the nitrocellulose filter with sodium dodecyl sulfate, the bacterial colonies will be lysed. After lysis of the bacterial cells that are present in the colony, next step is to treat with sodium hydroxide or is to treat the lysed colonies with a suitable alkali, for example sodium hydroxide. Treatment of the lysed colonies with sodium hydroxide will cause denaturation of DNA, so that DNA will be converted into single stranded form. After treatment with SDS and after treatment with sodium hydroxide, next step is to bake the nitrocellulose filter at 80 degrees centigrade. Alternatively, the nitrocellulose membrane that contain denatured DNA molecules can be exposed to UV light. So the baking at 80 degrees centigrade or by UV exposure will cause the fixation of the DNA molecules that are present in these colonies to the nitrocellulose membrane. So by baking at 80 degrees centigrade, the DNA molecule which are single stranded and which are present in these colonies, lysed colonies, they will fix on the nitrocellulose membrane. After fixation, next step is to treat this nitrocellulose filter with radioactive probes. Probes are short oligonucleotides that are radioactively labeled, or they can also be labeled by fluorescence. If the probes are radioactively labeled, then they can be detected by autoradiography. So the nitrocellulose filter that contain the fixed DNA is treated with suitable radioactive probes. So the probes will bind to the complementary DNA wherever they are present. After hybridization with a suitable radioactive probe, next step is washing. By washing, we can remove the unbound probes or excess probe can be removed from the nitrocellulose filter by the process of washing. So by treating this nitrocellulose filter with radioactive probe, hybridization occurs between the specific DNA that is present in the bacterial colony with the radioactive probe. So we can easily visualize that which bacterial colony contain the recombinant DNA or which bacterial colony contain the specific gene. That is the main objective of colony hybridization. So after treatment with radioactive probe, next step is autoradiography. So when the nitrocellulose filter is subjected to autoradiography by using a X-ray film, the bacterial colonies that contain the recombinant DNA or that contain the specific DNA will be visualized as black dots. These two black spots indicate that which bacterial colonies contain the recombinant DNA. After autoradiography, the nitrocellulose filter is compared with the master plate. So by comparing the nitrocellulose filter with the master plate, we can easily identify as to which bacterial colonies contain the gene of interest. So friends, this is about colony hybridization. Colony hybridization is a blood analysis technique in which bacterial cells are transferred from a solid nutrient medium onto a nitrocellulose membrane for further analysis. It is very simple but powerful technique useful for screening gene libraries. So friends, this is about colony hybridization. Please share, like and subscribe. My YouTube channel is Sam Online Class. Don't forget to give your feedbacks. See you, bye bye.